Professor João Pedro De Magalhaes from the University of Liverpool to talk about the genetics of aging.
Thank you. Thank you for the invitation. Let me try to share my screen. Okay, can you see my slides well? I take that as a yes.
Yeah, we can see you. Thank you.
All right, great, thank you. So thank you for the invitation. It's a pleasure to tell you. I mean, what I'll do is I'll provide a brief overview of really how much we've advanced in our understanding of manipulations of aging, particularly at the genetic level. I'll briefly touch upon environmental manipulations as well. I guess since the beginning of time that people have tried to overcome aging, trying to discover the elixir of youth. So I think this idea that we can reverse or at least slow down aging, or this dream has been embedded in humankind since for thousands of years. Now, what I hope to, and although that so far has failed, what I hope to convince you today is that we have now developed technologies and knowledge to, well, maybe not immortality, but at least to retard aging, which, as I'll tell you about, we can do in animal models to a very impressive degree already. Now, I think it's important, although we've heard about, and there are a number of theories of aging and hypotheses related to aging, that we still do not understand well the driving mechanisms of aging. So there's various hypotheses, cellular senescence we've just heard, DNA damage, but we still don't really know for sure what is the driving mechanism of human aging, or the driving mechanisms of human aging, in spite of these various hypotheses. Having said it, there is reason to be optimistic that we can intervene in aging. There's several lines of evidence of this. One of the things that has always fascinated me is the fact that there are species that appear not to age at all. And these are some of these examples. These are complex animals, complex vertebrates, as you can see. So this is a kind of fish called, kind of rockfish from a genus called Sebastes. These are painted turtles, this is Galapagos tortoises. This is a kind of blind cave salamander called the hole. So these are all complex animals that appear not to age. In other words, they don't exhibit the typical physiological functional decline that you see in humans and in, as far as we know, all mammals. And I've always thought of this as inspiration, a little bit like the Wright brothers took inspiration from birds, heavier than air animals that can fly. We can take inspiration from these animals because it shows that aging is neither inevitable nor universal. Natural selection can, at very least, result in animals, in species that age much, much slower than human beings. So, I mean, apparently it is not age, maybe they age after hundreds of years, we don't know. It's difficult to get grants for 5-year projects. But at the very least, they age much slower than human beings. Now, the other reason to be optimistic that we can intervene in aging and that we can manipulate aging comes from a number of studies as well in animal models. And these have been driven by a number of technological breakthroughs. One of them is at the level of DNA sequencing. So as I'm sure you're aware, the cost of sequencing has declined dramatically in recent years. So this is a log scale, and DNA sequencing is, you can now sequence your genome for less than £1,000. So this is really one of the technologies that's really allowed us to probe into some of the potential genes that modulate and regulate aging across species and within species. So I'll show you a couple of examples, first from long-lived species and then from model organisms as well. So, I mean, some of the work we've been doing is actually been focusing on long-lived disease-resistant species. One of these species is this animal called the naked mole rat. we're interested in these animals not just for their good looks. We're interested in them because they're rodents. They're quite small. They're a little bigger than a mouse. They're smaller than a rat. And they can live over 30 years, nearly 40 years, in fact. And they're very highly resistant to cancer. So this is one example of the species by which natural selection has, compared to mice and rats, allowed them to live much longer with a disease resistance. So we've been doing DNA sequencing in this species. I won't go into details on that. The other species we've been focusing is the bowhead whale, which is estimated to live over 200 years using biochemical assays. And that makes us the longest lived mammalian species that we know of. And what's quite remarkable about these animals is that, first, they live very long in a natural environment without doctors, without medicine. And particularly in terms of cancer, if you think about cancer as just one rogue cell that just proliferates uncontrollably, then these species that have 1,000 times more cells than humans, everything being equal, should develop cancer early in life. But they don't, or they would die of cancer early in life. So they wouldn't live so long. So they must have natural mechanisms to protect against diseases like cancer that we human beings lack. And so without going into great details, we've been trying to discover what are the genetic and molecular secrets of the species like the naked mullet and the bowhead whale. Just to give one very quick example, you can find specific changes in proteins and in genes that perhaps confer longevity and disease resistance. So this would be an example. So in green here, you see this is a protein called PCNA, proliferating cell nuclear antigen, which is involved in cancer and cell proliferation. And we find some specific changes in well-conserved positions in the bowhead whale, which is what you see here in red. And then we did this 3D models, which allow us to predict that these changes probably affect the interactions of this protein PCNA with another cancer-related protein called FAN1. So we think some of these changes that we identify in this long-lived disease-resistant species could be related to their longevity and their disease resistance. Now, of course, this is a computational model. I mean, we can't really do genetic knockouts or genetic manipulations in whales. We don't know for sure if these are involved, but it provides insights and clues into potential mechanisms and genes involved in longevity and disease resistance, which I see as a complementary way of doing biomedical research. While most scientists do research on short-lived mammals or short-lived organisms, we tend to look into long-lived disease resistance organisms. And ultimately, what I would like is for findings from these long-lived disease resistant organisms to apply this to human beings, either by developing pharmacological approaches, drugs that mimic the effects of the mutations we see in long-lived species, or even thinking further ahead, even via gene therapy, ex vivo gene therapy, in viral gene therapies. So that's something that more to the future. But that's the ultimate goal, is to try to gain insights from this species that we can then apply to live longer, healthier lives. And we know that there is a lot of possibilities in that context from animal models. So we know aging is surprisingly plastic in animals. I guess this started particularly at the genetic level in 1988. Tom Johnson and colleagues identified the first genetic variant that has a significant impact on the longevity of worms, which are, as you can see here, they're these tiny microscopic animals that live, normally they live only a few weeks. So this really started, well, this was the first gene, we didn't know at the time what the function of the gene was, so they called it H1, but it was the first single gene that we knew that had a significant impact on the aging and longevity of animals. There's been a lot of development since then, but the current record is about a 10-fold life extension. So again, a single genetic variant, interestingly enough, it's also in H1. It's a different genetic variant, but it's a single genetic variant that allows animals to live, instead of a few weeks, allows them to live for several months. So it's quite impressive how you can manipulate longevity now in these simple organisms. And then, of course, okay, so that's worms. What about mammals? in mammals, we also know that aging is surprisingly plastic. We can extend lifespan in mice up to about 50% by manipulating a single gene. And it's important to emphasize as well that we're not just extending the lifespan of the animals, we're also extending the health span of the animals. So long-lived mice, there's various of them, but in general, they tend to also be healthier for longer, which is what we want. If we want to apply these technologies, if we want to apply these discoveries to humans, that's what we want. We want to have a 70-year-old with the health of a 50-year-old. And at least in animal models, that's possible, which I think is quite a remarkable discovery, both from a conceptual perspective, that the process of aging is plastic, and from a practical perspective as well, that maybe we can apply these discoveries to human beings. So it's something actually I've been fascinated for a while. I mean, one of the things I studied years ago is this database called GeneAge Database of aging-related genes that includes all of these model systems and these individual genes that impact on aging. So you need to have a particular genetic manipulation, overexpression, mutation, silencing of a gene, and then if it has a significant impact on aging and longevity, then we include it in the database. And as you can see in worms, the current record is 10-fold life extension, and there's nearly 900 genes in worms that significantly impact on longevity. And for the various model systems, we now know over 2,000 genes that impact on longevity. So, and even in mice, we have over 100 genes that we can individually manipulate, and that they can either accelerate aging or retard aging. So just to give a very quick example, so these are two mice of roughly the same age. The animal on the right has a mutation, I think it's ERCC1, which is a gene, a protein involved in DNA damage responses. And when you mutate it, you have these animals that have a shorter lifespan, and they also look older than animals of the same age that don't have the same but well-typed controls. So we can at least apparently accelerate aging in animal models, and we can also retard it. So just to give one example, again, these are two animals of the same age, but the animal in the bottom has a mutation in growth hormone signaling, which, as the name implies, makes animals and humans grow. So you tend to have these cute little dwarf mice that also tend to be disease resistant, and they tend to be long-lived. And again, they still develop age-related diseases, but they develop them later in life. So really, I think these advances in genetics of aging have been quite remarkable for the field, showing that aging is a surprisingly plastic process. And to a certain degree, to certain limits, we can accelerate and retard aging in animals. Now, okay, so that's animals, but what about human beings? We know that there is an impact of genetics in human longevity. One example would be Jean-Claude, she's the oldest human being on record. She lived to be 122. And interestingly enough, she was very fond of port wine from my hometown of Porto. She ate massive amounts of chocolates as well, and she smoked for most of her adult life. She only stopped smoking because she was her eyesight was getting worse, and she was afraid that she would hurt herself with a cigarette. So another example of a healthy lifestyle you'd associate with the longest human being on record. And we know she's not an only one. When you look at centenarians, they don't tend to have lifestyle factors in common. Some of them, they care a lot about their health, have a healthy lifestyle, others don't. However, very frequently, longevity runs in the family. They tend to have other relatives that maybe were also centenarians or non-centenarians, so they tend to have other cases of long-lived individuals in their family. And you can actually study, the heritability of longevity in twins and families. And we know that there is a genetic component to human longevity, in particular to exceptional human longevity, like centenarians and like people like Jean-Claude. So I usually say that if you want to live 120 years, you have to choose your parents and grandparents well, because it's mostly down to genetics. Now, unfortunately, we still don't understand well, we still don't know which are the genes involved. So although there's been a lot of findings in the genetics of aging in animal models. A lot of discoveries, I mentioned over 2,000 genes we know in model systems that impact on aging. We actually know very little about the genetic basis of human longevity. We know there's a genetic basis, but we don't know yet which genes are involved. So that's still, I think, a big gap in our knowledge. So I guess I hope I'm persuaded that it is possible to manipulate aging. Aging is not just a collection of diseases running in parallel. Now, we cannot, at least not yet, manipulate genes in human beings the way we can use genetic engineering to manipulate them in animals. But potentially, we could manipulate genes by diet and pharmacological interventions. And if we could then translate these findings from model systems in humans, it would have an unprecedented impact on human health. And indeed, we do know that there is an impact as well of diet on aging. We know that from animal models. Again, without going into great details, we know that there's dietary manipulations like caloric restriction. So caloric restriction consists of reducing the amount of calories while keeping other vitamins and nutrients healthy. And we know that in animal models like mice, worms, and flies, it extends lifespan and it retards the onset of age-related diseases. In some strains of mice, it can have an impact of up to 50% increase in lifespan, which is quite impressive and equivalent to what you see with genetic manipulations. So we know that dietary manipulations can also have a significant impact on aging, at least in animal models. Whether they translate to humans, that's still an open question. The other aspect, well, the other angle that's been rapidly growing is the level of longevity pharmacology. So we now know of various longevity drugs. We know of various drugs like rapamycin and metformin. So what you see here is actually rapamycin. We know of various drugs that retard aging in animals, that extend lifespan. I would say that there is a difference. I think longevity drugs, they tend to have smaller effect sizes than genetic manipulations. So it's more around 10%, 15% maximum life extension you see from pharmacological interventions in mice, while genetic manipulations, well, it's up to 50%. So you tend to have greater effects from genetic manipulations, not surprisingly, because changing a gene is a bigger biological change than giving a drug. So, but still, when we're thinking about applying this knowledge to human beings, I do think this is one of the ways that we can translate findings from these basic discoveries in biology and genetics of aging all the way to human beings. Now, I'm not the first one to say that or to think of that. I think that's one of the goals of the field, really, is to develop interventions for aging. So it's something I think the field has been focused for many years, but really these developments in the genetics of aging and the manipulations of aging have really, I think, given new impetus and have fostered really a number of translational approaches. And perhaps in that context, and not surprisingly, I think there's one of the big changes I see in the field of aging and longevity from when I started over 20 years ago, is that you see this rapid and impressive growth in longevity biotech. So I think arguably Calico, the company that Google started some years back, that was on the cover of Time magazine, arguably they're the most famous company in the space. But there's various others, like Unity Biotech, Juvenescence. You might have seen the news that Jeff Bezos is also investing in this new enterprise called Altos Labs, focused on rejuvenation technologies. So we have this very impressive and rapid increase in terms of longevity biotech, which I think is a very healthy reflection of the field, of the fact that there's science-driven opportunities in the manipulation of aging that investors and companies see as potentially having a human impact and therefore having commercial interest as well. So I think I'm running out of time, so I'll to summarize, I I would say that aging has a major impact on societies and health, something we've heard about in this conference already, but it remains a mystery of biology. We still do not understand what are the molecular mechanisms driving the process of aging, even though there's various hypotheses. I told you about the studies of lonely species to find genes that protect against aging and disease. Again, it's complementary to traditional biomedical approaches. Then at the genetic level, I told you about these major breakthroughs that in the past 30 years of the genetics of aging in animal models, how we now know that genes regulate aging in animals and can manipulating one single gene can have significant impact on longevity and aging phenotypes in animals. I told you that human longevity has a genetic basis, but we still don't yet understand, or we still don't know which are the specific genes involved. And I told you then going forward, really there's the potential for translation, this plasticity of aging that we observe in animals to humans, in particular by dietary and pharmacological manipulations. And not surprising, I think, again, a reflection of how much the field has progressed. We have this rapid growth in longevity biotech, hoping or aiming to capitalize on this scientific advances and hopefully to develop. I am fairly optimistic about the development of therapies, in particularly pharmacological longevity drugs. I am fairly optimistic about that this will come to fruition in the next 5 or 10 years. So with that, well, thank you very much for your time and attention. I mean, this is our lab pre-pandemic. I didn't really talk all that much about what we do, but if you have any questions, happy to answer. Thank you very much. Thank you.
Unfortunately, we don't have any time for questions, so thank you very much. And I'd like to call on Professor Avram for some closing remarks.
Thank you, Danielle. She normally calls me Karen. Okay, wow, that was wonderful. I'm a geneticist, so that was a great finish for me for the day. And then the other thing is that I opened up my introduction in the morning talking about these companies, so I think we've come full circle. So, for those of you who are still here, you are the most serious about aging. I'm not sure what the context that is. Please save your name tags. Bring it with you tomorrow morning when you come in. We start the same time tomorrow. Very comfortable for Israelis, at least 10:30 in the morning. And thank you. It was a phenomenal day. I really enjoyed it, learned an enormous amount. I hope you did as well. For those of you who are joining for dinner, we're going to meet out in the front. There's a bus taking us. Those of you who are parked inside the university, don't worry. I will get you to your car afterwards. You're not going to be stranded at Tel Aviv University for the night. Have a wonderful evening, everyone, and see you tomorrow morning.